Hi there, AP biologists. This video is about gene expression in prokaryotes, or in other words, how do bacteria control how their DNA gets translated and transcribed into usable proteins. They do this using a system called the operon. An operon is a unit of DNA that contains a cluster of genes under the control of a single promoter. So here is an operon. We can have the operator, and then in this case it shows 3 genes. Ahead of the operon is a promoter. This is where the DNA polymerase binds to transcribe the DNA into messenger RNA. The operator acts as the on-off switch. When the operon is in the on position, those genes get transcribed into messenger RNA, which then get translated into separate protein products. Now, the operon system can be turned on or off in a number of different ways. We've got negative control and positive control. In negative control, a repressor protein is going to turn the operon off. So normally, the RNA polymerase would bind to the promoter, move through, read the information on the DNA to make a strand of messenger RNA and therefore, and then later on down the road, proteins. However, with negative control, a repressor protein is going to block and bind to the operator, which blocks the RNA polymerase, blocking transcription, turning the gene off. With positive control, we have the opposite. A protein or an enzyme is going to bind to the promoter, which actually enhances RNA polymerase's binding ability, and then we transcribe and then can translate the gene. So let's take a look at the 2 types of negative control, and then we'll take a closer look at positive control as well. We have 2 types of negative control. The first is an inducible operon. This operon is usually in the off position, and we have an active repressor protein that's binding to the operator, preventing transcription from occurring. But if an inducer molecule comes along and binds to the repressor protein, it changes its shape, and the repressor protein is inactive and can no longer bind to the operator. Once it separates itself from the operator, the RNA polymerase is able to move through, reading the DNA nucleotides and transcribing the messenger RNA. Let's take a look at a specific example that we actually see in bacteria cells. Here we have what's called the Lac Operon. In this operon, we have 3 genes that normally code for the production of 3 different enzymes, and those enzymes are responsible for breaking down lactose, which is a component that you find in milk. So, but normally bacteria are in an environment where there is not lactose present. So it would be really a waste of their resources to continue to transcribe and translate these genes to make enzymes that they can't even use. So normally this gene is in the off position because we have a repressor protein that's bound to the operator. However, let's say you take a big drink of milk or eat some ice cream, and all of a sudden you've introduced a bunch of lactose into your stomach where all of these bacteria are living. In addition to introducing a bunch of lactose, you're also going to introduce another similar structured molecule called allolactose. This allolactose also acts as an inducer, and it binds to the repressor protein, changing its shape and removing it from the operator. Well, now the RNA polymerase can go through and transcribe and translate the gene, making the messenger RNA and all of these enzymes. Now those enzymes can be used to digest the lactose and the allolactose that's now found in the bacteria's environment. Eventually, it'll break down enough of it to, and those inducers will be removed from the repressor protein, and the repressor protein will bind again to the operator, preventing transcription, and we stop making those enzymes because they're no longer needed. So it's a pretty neat little system. It turns itself on and off when appropriate. Another example of a negative control operon is the repressible operon. These operons are usually in the on position, so the repressor protein is inactive and transcription is occurring. It's making proteins, messenger RNA, and then therefore down the road proteins. Now, a co-repressor molecule is going to bind to the repressor, changing its shape, activating it, and then it can bind to the operator, which then blocks the RNA polymerase from transcribing the gene, and we no longer make those proteins. A really great example of this is the Trp Operon system in your body, or in bacteria. So normally, bacteria are going to have this operon turned on. RNA polymerase will be transcribing the messenger RNA, and we're going to make a bunch of proteins which are enzymes that are used for the synthesis of the amino acid tryptophan, which bacteria normally don't find in their environment. So they have to make it themselves, which is why this gene is normally turned on. However, if they get into an environment where there's plenty of tryptophan, or they've made enough tryptophan, the tryptophan actually acts as a co-repressor molecule, changing the shape of the repressor, and that repressor binds to the operator, preventing RNA polymerase from binding and transcribing the gene. So we no longer make all of those enzymes that build the tryptophan. Over time, that tryptophan goes away, and then we can turn the gene back on. So again, a neat little system. Next up, we have positive control of those genes. So in addition to being able to turn the genes off using a repressor, we can also activate those genes using positive control. So down here, you can see that little purple Pac-Man shape. That's our representing our inactive activator protein. And then we can also have a ligand. Right now, the RNA polymerase is not able to bind to the promoter. It needs help. And in order to help it, we have to activate this activator protein. So it binds then to the promoter, which helps the RNA polymerase bind, and then it can translate and transcribe those genes. So that's really it for the Operon system or the prokaryotic gene expression. It all depends on whether the gene is turned on or off, whether we make those proteins. I hope you found this helpful.